Hi there. Today I'm going to be talking about embryonic stem cell research, scientific and religious views, and the cure of disease. Table of contents. First, we're going to look through the introduction, and then in vitro fertilization. Then we'll go through stem cell basics, and then learn a little bit about adult stem cells. Then we're going to look at two case studies from families. Then we're going to look at politicians and embryonic stem cell research. We'll look at the Catholic Church on what they think about the embryonic stem cell research. Then we'll go into the Christian view on the embryonic stem cell research. And then, and I don't have it mentioned here on the table of contents, but we're going to look at, what are the views around the world about the embryonic stem cell research, and then at the end we'll just finish off with a conclusion and then my references. And introduction. Stem cell research has enormous potential when it comes to the advancement of medicine. It has the ability to create new organs, tissues, and systems within the human body. Cursor 2004 explains as an example of how a batch of stem cells might be reconstructed into cardiac muscle cells and injected into an injured or diseased heart, thus repairing the damaged muscle. Stem cells could one day possibly treat previously incurable devastating conditions such as stroke, certain types of cancer, leukemia, lymphomas, lung cancer, muscular dystrophy, Alzheimer's disease, Parkinson's disease, diabetes, heart disease, and spinal cord injuries. Scientists have used them to screen new drugs and to identify the cause of birth defects by developing model systems. However, Steinbach, 2007, affirms that the dilemma arises when embryonic stem cells are derived from embryos that have been fertilized in a woman's body. Some see the destruction of an embryo as the murder of an unborn child, and it is in this area of science which raises many ethical debates. In vitro fertilization. When a sperm fertilizes an egg cell, it divides and becomes an embryo. In the embryo, there are stem cells that are capable of becoming all the various types of cells of a human body. Scientists get embryos in two different ways. The first method is with in vitro fertilization. A couple's sperm and eggs are fertilized in a culture dish. The eggs are developed into embryos, which are then implanted in the female. Nevertheless, more embryos are created, thus can be implanted, and the remaining embryos are usually frozen. Many couples donate their unused embryos for stem cell research. The second method is through therapeutic cloning. This technique merges a cell from the patient who needs the stem cell therapy with a donor egg. The nucleus is removed from the egg and replaced with the nucleus of the patient's cell. This egg is stimulated to divide either chemically or with electricity, and the resulting embryo carries the patient's genetic material, which significantly reduces the risk that his or her body will reject the stem cells once they are implanted. Both methods use existing fertilized embryos and create new embryos for research. Stem cell basics. Sills 2005 explains that an embryo contains about 4 to 8 egg cells. These stem cells are called totipotent and can develop into all types of cells. At 3 to 5 days, the embryo develops into a ball of cells called a blastocyst. A blastocyst contains about 100 cells in total, and the stem cells which are inside are pluripotent and can also develop into almost any type of cell. To grow the stem cells, scientists remove them from the blastocyst and culture them, so they grow them in a nutrient-rich solution in a petri dish in a laboratory. The stem cells divide several times, and scientists divide the population into other dishes. After several months, there are millions of stem cells. If the cells continue to grow without differentiating, then the scientists have a stem cell line. Cell lines can be frozen and shared between laboratories. Stem cells serve as a repair system for the body. They have the potential to develop into many different types of cells in the body. Stem cells are different from other cells in the body in 3 ways. They can divide and renew themselves over a long time. They are unspecialized, so they cannot do specific functions of in the body, and they have the potential to become specialized cells such as muscle cells, blood cells, and brain cells. There are two main types of cells: embryonic stem cells and adult stem cells. Embryonic stem cell research looks for ways to use stem cells for regenerative or reparative medicine. These stem cells are thought to be the key that will unlock the cure to many diseases, from Alzheimer's to rare immune and even genetic disorders. Adult stem cells. Adult stem cells are built in repair kits, regenerating cells damaged by disease, injury, and everyday wear and tear. However, Cruiser 2004 explains that adult stem cells are much less promising than stem cells that are extracted from embryos, only giving rise to the same type of tissue from which they originated. Like embryonic stem cells, adult stem cells can be grown in culture to establish stem cell lines. Stotz et al. 2006 mentioned that unlike embryonic stem cells, adult stem cells do not present ethical problems. Case study: Molly and Adam Nash. McGill 2002 described the story of Molly Nash, an only child of 6 years of age with a rare genetic disorder called Fanconi anemia. Not only does the disease prevent the production of bone marrow, but can kill at a very young age. However, a bone marrow transplant can offer an 85% rate of success in treating the disease. But there's a catch. It must come from a sibling. With no brother or sisters, the parents decide to have another child. Their hope is to use blood from the placenta and umbilical cord once the baby is born for a stem cell transplant for Molly. The parents opted for assisted reproduction. In addition, they had pre-screened the baby using pre-implantation genetic diagnosis to make sure it did not have the same disease as Molly. Baby Adam was born and Molly received a transfusion of stem cells from the umbilical cord and placenta. Both babies have flourished. The ethical controversy arises with the 15 human embryos that were created via in vitro fertilization. Case study: Andy and Sofia Trevino. Parents Tanya and Andres found out that after the birth of their son Andy, he had a disease called Nemo syndrome, and it's a rare genetic disease. That impacts the immune system, causing frequent infections that are life-threatening. Doctors recommend a bone marrow stem cell transplant to try to replace parts of the immune system and help him fight the infections. Nemo-free stem cells from the bone marrow could help him fight the infections. However, after 2.5 years, there was no match that could be found. So they decided to have another baby in vitro fertilization and use the baby's umbilical cord stem cells for Andy. It took 5 in vitro fertilization cycles and 36 embryos for Sophia to be born. Even though stem cells were taken from her umbilical cord, it wasn't enough. They waited 6 months until baby Sophia could donate more stem cells, this time taking it from her bone marrow, and after about 1,000 days in hospitalization, Andy was cured. Politicians and stem cell research. Stadelman and Torgler 2017 point out that embryonic stem cell research has been hotly debated among politicians. However, it has never been fully banned in the United States, but rather restrictions have been imposed on funding and the use. The authors explain that in 1995, President Bill Clinton permitted federal funding for research on embryonic leftover from in vitro fertility treatments, and also recommended federal funding of research on embryos specifically created for experimentation. However, in 2001, U.S. President George W. Bush restricted stem cell research, which led to the legal uncertainties that have affected its use in the U.S. But in March 2009, Barack Obama removed certain restrictions on federal funding for researching involving new lines of human embryonic stem cells. Prior to President Obama's executive order, federal funding was limited to non-embryonic stem cell research and embryonic stem cell research based upon embryonic stem cell lines in existence prior to August 2001. The National Conference of State Legislators, an organization in the U.S. that fosters interstate cooperation and facilitates the exchange of information among legislatures, specified that several states restrict and may have complete ban on embryonic stem cell research in the U.S. And a few examples, and there are many more states where experimentation on any live fetus is prohibited, are states of Florida, Kentucky, Louisiana, Iowa, Texas, Maine, Michigan, and Montana. However, such states as California, Illinois, Connecticut, Indiana, Iowa, Massachusetts, New York, and New Jersey allow experimentation on any live fetus embryo. A Catholic defense on embryonic stem cell research. Whittington 2012 states that the Catholic Church is greatly opposed to embryonic stem cell research because this type of research destroys embryos, which the Church deems as full persons with all basic rights of other human persons. It is seen as intrinsically evil. However, the author argues that there are limited circumstances in which parents may donate embryos to establish stem cell lines that respect the rights of the embryonic person. First, ova are obtained and fertilized for use in IVF with the intention of transferring all the embryos for gestation. Second, the numbers of ova that are obtained are sufficient to have reasonable probability of producing the number of pregnancies that the couple had decided to pursue, but not so great as to anticipate a significant likelihood of surplus embryos. Third, the couple completes the childbearing or the biological mother is biologically unable to become pregnant. Fourth, the couple is willing to offer the remaining embryos to other couples, but there are no available recipients. Fifth, there is no organization willing to take custody of the embryos and screen prospective parents. Sixth, the parents voluntarily designate the embryos for donation for embryonic stem cell research, and stem cell donation is conducted in accord with dead donor rule. Seventh, the embryos are thawed and lose viability using the routine procedure of fertility cleansing for disposing of surplus embryos. 8. When the embryos are determined to be non-viable by the fertility center, they are transferred to research scientists for the isolation of pluripotent stem cells. Cryopreserved embryos cannot be maintained indefinitely, so if parents aren't able to gestate the embryo and there is no other option such as adoption, the embryo is stuck in a permanent state. Therefore, it cannot develop into a person. There is no real moral obligation to continue care, and the embryo can die. Christian ethics on embryonic stem cell research. Geisler 2010 states in the chapter on biomedical issues from his book Christian Ethics, Contemporary Issues and Options, 2nd edition, that from a Christian perspective, stem cell cures for the use of embryonic stem cells is unacceptable for the same reason abortions are not acceptable. It takes away a life. Geisler argues that it violates several ethical principles from a Christian viewpoint. First, that just because we can do something doesn't mean we should do it. Second, the end does not justify the means. Third, we should not take a life to lengthen a life. The author gives the example that even though death row prisoners are going to die anyways, it does not give us the right to use them as sacrificial medicine in the name of science and progress. Geisler mentions that even though couples are successful in conceiving a child through in vitro fertilization, he questions whether it should be done. His main concern is the wasted embryos from the process and the fact that the majority of embryos are being sacrificed for one that will survive and prosper. Since the end doesn't justify the mean, Geisler stands firm when he states that Christianity sees in vitro fertilization and embryonic stem cell research as morally wrong. Views around the world. Savaraman and Noor, 2006, explore multi-faith ethical viewpoints from Buddhist Hindus and Catholics in Malaysia on embryonic stem cell research. Their studies found that Hindu subjects believed that the use of surplus embryos had less moral weight attached compared to a research embryo, which is specifically created for the sole purpose of research and not part of an infertility treatment. Buddhist subjects did not see any ethical difference between the use of either source of embryos as long as embryonic stem cell research was done responsibly. As mentioned before, the authors found Catholic subjects opposed to the use of both surplus or research embryos. In Malaysia, Islamics allow embryonic stem cell therapy if conducted on surplus embryos for maintaining well-being and relieving people's suffering. However, the use of research embryos is prohibited. Wing 2008 states that China has several research centers with few barriers when it comes to human embryonic stem cell research. Pew Research 2008 stated that in South America and Brazil permit stem cell research using excess in vitro fertilized embryos that have been frozen for at least 3 years. Mexico is known for its stem cell research and often treats chronically ill foreigners with cerebral palsy, diabetes, autism, brain and spinal conditions, and heart disease. Research is permitted in Canada on discarded embryos from in vitro fertilization procedures. Nonetheless, it is against Canadian law to create human embryos for research. Belgium, France, Sweden, Israel, India, and South Africa forbid reproductive cloning but allows therapeutic cloning. The United Kingdom leads the race in stem cell research in Europe. And because of unethical medical experiments conducted by the Nazi regime during World War II, Germany has one of the most restrictive policies for human embryonic stem cell research. Conclusion. The debate on using embryonic stem cell research is greatly dependent on what you think embryos are. If they are seen as full persons from conception, then it is morally wrong and basically murder. If life is not defined at conception, then using embryos that were created for couples with reproductive issues that would be discarded is a great opportunity to research disease and diminish human suffering. So there is often no in-between views. And what does the future hold? Well, synthetic stem cells seem to be promising. And in 2007, a team of scientists from the North Carolina State University was able to implant synthetic cardiac stem cells which repaired muscle tissue that got damaged by a heart attack. And that's about it. And the next 2 slides are actually my references for my work. Thank you.